In this video, we'll explore what live blood analysis is and examine the impact the atom cell device had during this type of analysis. Live blood analysis is a screening method that allows us to observe the various components of a fresh blood sample under a microscope. It provides insight into the functionality of the blood system and can reflect aspects of a person's overall health. Human blood is made up of approximately 45% cellular components, namely red blood cells, white blood cells, and platelets, and 55% plasma. Blood performs many vital functions, including transporting oxygen, regulating body temperature, and supporting the immune system. The condition of the blood often correlates directly with our overall health and can be influenced by diet, lifestyle, and environmental factors. During a live blood analysis, a small drop of blood is obtained using a finger prick. The sample is placed directly onto a glass slide and viewed immediately under a microscope at up to 1,000 times magnification. The lighting technique used allows even the smallest details of the blood to be seen clearly. This method enables practitioners to observe the structure and behaviour of the blood components in a near-living state, similar to how they function inside the body. The atom cell is designed to be worn on the body to help protect the wearer from the potential negative effects of background electromagnetic radiation. To assess its effectiveness, we conducted a live blood analysis study using a smartwatch as a radiation source. The goal was twofold. First, to determine whether exposure to this device affected blood health, and second, to evaluate whether wearing the atom cell on the body could influence the outcome. In the second phase of the study, a new baseline blood sample was taken from the test subject. The subject's food and fluid intake had been carefully monitored and no smartwatch had been worn prior to testing. The initial sample revealed a healthy blood picture. Red blood cells were freely flowing, cell membranes were intact, and the plasma was clear. Next, the test subject wore an active smartwatch for 20 minutes. 10 minutes after removing the device, a second blood sample was taken. This sample showed a noticeable decline in blood quality. Red blood cells were clumping together and blood flow appeared restricted. Following this, the subject wore the same active smartwatch for another 20 minutes, this time while also wearing the atom cell. 10 minutes later, a third blood sample was taken. The results were striking. The third sample showed a significant improvement in the blood profile, even exceeding the quality observed in the baseline sample. Red blood cells were no longer clumped together, rouleau formation was absent, and blood flow had been restored. The images from the analysis suggest that the atom cell may play a protective role against the adverse effects of electromagnetic radiation on blood health. The full data set from this study is available for viewing on the research page of atomlab.uk.